Hi, you know what? It's always better to be in thirsty knowledge. But what you also should know is anything in excess is not so good even if it's your life. Knowledge is also like that. If you know too little, then the life is difficult. And if you know too much also the life is difficult. But knowing exactly what you need to know gives you the best life you can imagine. But you should choose it wisely. So in next 3 minutes, I will tell you what exactly you need to know about PCR tests, which you happen to hear more often as a result of Covid-19 pandemic. The PCR refers to polymerase chain reaction. Here polymerase is an enzyme required to reproduce DNA. Chain reaction means a repetitive work. So frankly, PCR is a method developed to reproduce DNAs. The purpose of amplifying DNA is to make it easy to analyze them in detail. DNA is a double-stranded molecule which is in the cell nucleus. Those DNA consist of all necessary instructions to produce required proteins in order to develop and reproduce organisms. DNAs are made up from 4 types of molecules called nucleotides. These nucleotides are building blocks like elements which are made from 3 things: a phosphate group, a sugar group, and one of 4 types of nitrogen bases. The 4 types of nitrogen bases are adenine, thymine, guanine, and cytosine. The order or sequence of these bases determines what biological instructions are contained in a strand of DNA. For an example, the sequence ATCG-TT might instruct for blue eyes, while ATCG-CT might instruct for brown. Anyway, these nitrogen bases have connected with their complementary bases with hydrogen bonds. Hence, the DNA double helix has formed by twisting around each other. I know, in order to learn about DNA precisely, there is a vast amount of knowledge you need to gain. But I'm not going to dive through all of them. I will tell you only what you need to know in order to understand PCR. Before PCR method was invented, it took weeks to clone a DNA. But after the Nobel Prize winner Kerry Mullis invented PCR method in 1983, it takes just a few hours to reproduce billions of DNAs. There are innumerable uses of PCR test. The one that we are mostly familiar these days is identifying COVID-19 virus infected persons. But there are so many other uses like find the paternity, study human evolution, clone DNAs of Egyptian mummies, DNA fingerprinting, and diagnose genetic diseases. To copy DNAs, PCR uses the same molecules that nature uses. DNA polymerase enzymes, which is the enzyme required to reproduce DNA. Primers. This is a very important short single-stranded DNA part. Without this, polymerase enzyme cannot start the reproduction. I will tell more about this later. Nucleotides. These are previously mentioned building blocks consist of sugar phosphate part and nitrogen base. Basically, PCR test is a repetition of an automatic thermal cycle. There are 3 steps. Denaturation. Initially, the test tube is heated to 95 degrees Celsius, at which the double-stranded DNA melts and opens into 2 pieces of single-stranded DNAs. Annealing. Here the temperature brings down to 54 degrees Celsius, at which the primers get attached with the initial nucleotides of DNA strand provided that the nitrogen bases match to each other. If the primers in test tube don't have the particular complementary nitrogen bases of the initial nucleotides, then this step won't happen. Let's just assume the primers and DNA strand match together in order to move forward. Then the next step is extension. Again the temperature is brought up to 72 degrees Celsius, at which the polymerase enzyme works best. So as the primers are attached, now polymerase can gather all required complementary nucleotides and complete the DNA. Now you can see that after this cycle, one DNA has become 2 DNAs. So in each cycle, amount of DNAs get doubled. But this occurs only if significant primers for the DNAs are provided. Usually, PCR tests run for cycle number of 30 to 40. If initial nucleotides in the DNA matched with the primers in the test tube, then billions of DNAs are reproduced within 3 to 4 hours. If primers don't match with the DNA strand, then no reproduction occurs. So let's talk about PCR tests relevant to COVID-19 virus. COVID-19 is a coronavirus. Corona is a Latin word which means crown. It has got that name because it has a crown-like appearance from outside, but from inside it consists of an extraordinarily long single-stranded RNA genome. For PCR tests, initially, the RNA molecule is extracted from the cellular material. But the issue being, PCR is only applicable for DNAs. because RNA strands are not stable in alkaline conditions in the test tube. Also, polymerase doesn't react with RNAs. So we need to convert this RNA to its complementary DNA known as cDNA using a procedure called reverse transcriptase. For this, a reverse transcriptase enzyme is used. This process occurs in 55 degrees Celsius. After this step, standard PCR procedures can be followed. This PCR test method is called RT-PCR. This is the same method used to identify HIV virus also. In order to collect samples, nasopharyngeal or oropharyngeal swab methods can be used. Then RT-PCR method is proceeded with the significant type of primers which will anneal only with Covid-19 complementary DNAs. With the number of thermal cycles, if the number of complementary DNAs amplifies, it means the tested sample was infected with the Covid-19 virus. The number of amplified complementary DNA can be monitored real-time using a fluorescent dye in the test tube. It emits a fluorescent signal as the PCR produced accumulates in the sample. If you have questions regarding this PCR test, such as how to make particular type of primers and how exactly real-time PCR works or any other doubt, let me know in the comment section. I will clear your doubt. Thank you for watching my video. Subscribe if you feel valuable to get in touch with my future videos. Stay safe and stay hungry for knowledge. See you in my next video.